interference, C. RNA splicing, D. DNA translocation. What do you think is the correct answer here? This ApoB48 is a remnant of RNA editing. What happens in liver cells? The ApoB100 gene gives rise to ApoB100 protein, but ApoB48 protein is made in the intestinal cells from the gene encoding ApoB100 by the process of RNA editing, which is also known as differential RNA processing or chemical modification of RNA. This is a post-transcriptional modification. RNA splicing is also a post-transcriptional modification, but this is a removal of introns and not editing. Then coming to RNA interference. RNA interference is silencing technique where the gene is silenced by not letting the product of gene form. Translocation occurs during translation. This is movement of ribosome over mRNA. So the correct answer here is A, RNA editing. Hepcidin decreases iron absorption by inhibiting A. Divalent metal transporter, B. Hepahestin, C. Ferroportin, D. Transferrin. So the correct answer here is ferroportin. This hepcidin regulates iron metabolism. It mainly regulates iron entry in circulation. By inhibiting ferroportin, hepcidin reduces dietary iron absorption. The iron release from macrophage is also reduced by ferroportin inhibition. So increased hepcidin activity is responsible for reduced iron availability. Increased hepcidin will lead to anemia. Decreased hepcidin leads to iron overload. Hepcidin synthesis and secretion by liver is controlled by iron stores within the macrophages, inflammation, hypoxia, and erythropoiesis. It is ferroportin. Let's move to the next question. Which of the following has the highest glycemic index? A. Glucose, B. Fructose, C. Sucrose, D. Sorbitol. Now, what is glycemic index? Glycemic index, there is an increase in the blood glucose after the test dose of a carbohydrate compared with that of an equivalent amount of glucose. Glycemic index is a value assigned to foods based on how slowly or how quickly those foods cause an increase in blood glucose level. The highest glycemic index is for glucose and galactose. Also, lactose, maltose, trehalose, and isomaltose gets converted to glucose, so they also have high glycemic index. Now, glycemic index of fructose is less than that of glucose. Fructose and sugar alcohols have less glycemic index because they are not absorbed completely. Sucrose has low glycemic index than glucose as it gets splits into glucose and fructose. So the correct answer here is A, that is glucose. Now GLUT5. GLUT5 is a transporter of A. Galactose, B. Fructose, C. Mannose, D. Glucose. This GLUT5 is a transporter of fructose. Which of the following is insulin dependent? A. GLUT1, B. GLUT2, C. GLUT3, and D. GLUT4. Only GLUT4 is insulin dependent. This insulin recruits the GLUT4. This GLUT4 is present on the membrane of peripheral tissue, that is skeletal muscle, cardiac muscle, and adipose tissue. It helps in insulin dependent glucose uptake after meals. So it is GLUT4. Cause of HHH syndrome, that is hyperornithemia, hyperammonemia, and homocystinuria, is A. Deficiency of ornithine transcarbamylase, B. Defect in ornithine transporter, C. Deficiency of ornithine aminotransferase, D. All of the above. Now, deficiency of ornithine transporter leads to HHH syndrome, that is hyperornithemia, hyperammonemia, homocystinuria. This prevents ornithine transport into mitochondria. Hence, if it is in the cytoplasm and its reduced ability to clear carbamyl phosphate and ammonia. Mitochondrial carbamyl phosphate carbamylate lyses to homocystine, leading to homocystinuria. So there is a defect in ornithine transport. Which method is used to separate a mixture of lipids? A. Isoelectric focusing. B. Page. C. Chromatography. D. Electrophoresis. It is chromatography. Wrinkled paper appearance of macrophage in bone marrow aspirate is seen in A. Tay-Sachs disease, B. Gaucher's disease, C. Niemann-Pick disease, D. leukodystrophy. It is Gaucher's disease. Wrinkled paper or crumpled tissue appearance of macrophage in bone marrow aspirate is seen in Gaucher's disease. In CRISP or Cas9 system, which repair mechanism is used for genome editing? A. Homologous pair, B. Non-homologous end repair, C. Mismatch repair, D. Nucleotide excision repair. So the correct answer here is non-homologous end repair. Rapport Lubering cycle shifts oxygen dissociation curve to right because of A 2,3 DPG, B 1,3 BPG, C 3 phosphoglycerate, D 1,6 fructose biphosphate. So the correct answer here is 2,3 DPG. Shifting of oxygen dissociation curve to right means decreasing the oxygen saturation of hemoglobin as shown in the graph. Hot flashes are experienced as a result of increased noradrenaline, decreased estrogen, increased noradrenaline and decreased estrogen, increased noradrenaline and increased estrogen. It is increased noradrenaline and decreased estrogen. Restriction fragment length polymorphism is used for A. Analysis of DNA fragment, B. DNA estimation, C. Synthesis of nucleic acid, D. Detection of protein in a cell. Well, it is analysis of DNA fragmentation. Thermogenic food is which of the following? A. High protein diet. B. High carbohydrate diet. C. High fat diet. D. It does not depend on macronutrient. It is high protein. Ketone bodies are not utilized by A. Brain. B. RBC. C. Heart. D. Skeletal muscle. It is RBC. Enzyme deficient in hyperammonemia type 1 is A. Ornithine transcarbamylase, B. arginase, C. Carbamyl phosphate synthase 1, arginosuccinate synthase. It is carbamyl phosphate synthase 1. Type of mutation seen in sickle cell anemia is A. Insertion, B. Deletion, C. Point mutation, D. French mutation. It is point mutation. Which replacement of amino acid does not alter in normal function? A. Glutamine replaced by arginine, glutamine replaced by alanine, glutamine replaced by glutamate, glutamine replaced by aspargine. It is glutamine replaced by aspargine. The clinical feature given shown is A. Tryptophan, B. Phenylalanine, C. Cysteine, D. Glutamine. What do you think is the correct answer here? It is tryptophan. What you can see is Casal's necklace. It is shown Casal's necklace, also shown vitamin. It is due to vitamin B3 or niacin deficiency. Niacin is found from tryptophan amino acid. So tryptophan deficiency can also lead to Casal's necklace. 60 milligram of tryptophan gets converted into 1 milligram of niacin. Vitamin B3 deficiency pellagra. It is characterized by 4 Ds of pellagra. They are dermatitis, diarrhea, dementia, and death. So the correct answer here is A. Tryptophan. Which of the following binds mRNA with ribosome in eukaryote? A. Polyetine tail. B. tRNA. C. 7-methyl guanosine cap. D. Shine diagonal sequence. It is 7-methyl guanosine cap. What happens in eukaryotic cells? The ribosome recognize most eukaryotic mRNA by binding to 7-methyl guanosine cap at their 5' terminus. Shine diagonal sequence is presented -10 position on mRNA only in prokaryote and is rich in purine. In smaller subunit of ribosome, 16S RNA is present. SD sequence is complementary to 16S RNA. Which of the following is anaplerotic reaction? A. Conversion of pyruvate to lactic acid, conversion of pyruvate to oxaloacetic acid, conversion of pyruvate to acetyl-CoA, conversion of pyruvate to acetyl-aldehyde. It is conversion of pyruvate to oxaloacetic. Which of the following group of protein assist in folding of other proteins? It is chaperones. Myoglobin has which structure? A. Primary structure, secondary structure, tertiary structure, primary, secondary, tertiary, and quad. It is primary, secondary, tertiary structure. Best in investigation of metabolic disorder is Western blot, B. Tandem mass spectrometry, CPCR, and D. Gel electrophoresis. Most powerful technique of screening for inborn error of metabolism is tandem mass spectroscopy, also called TMS. So the correct answer is B. Separation of protein by their mass is by A. Gel filtration chromatography, B. Ion exchange chromatography. It is gel filtration chromatography.